All right. Good afternoon, guys. Can I get some quick acknowledgement that you can hear me and that you're seeing this screen?
Yes.
Thank you, Lisa. All right, cool. So as we discussed on Tuesday, you guys voted to have today's class dedicated to Unit 2 review as we are approaching the Unit 2 lecture exam next Wednesday. So this is going to be fully guided by your questions or topics that you want to spend time discussing. So if you guys came with questions prepared, let me know. Where should we start? Any questions?
Anybody didn't ask, so it's not a question, but if you have time to explain for me a little bit about action potential, if is that okay?
Yeah, absolutely.
Okay.
Let me see if I have a good slide to just address that. If not, we can talk about a video of it. I'm going to try using a whiteboard on here. Harder than using a whiteboard in person, but we're going to try to make this work. Okay, so essentially, we've got a membrane. Say this is outside. This is inside. And then let's draw some proteins in here. Okay. So throughout our cell's membranes and all of our cells, we have a lot of membrane-bound proteins that act as channels for things to flow through. And so an action potential is all about the electrochemical gradient of this membrane. So we're basically going to be addressing electrochemical gradient. So the electrochemical gradient is considering two parts. We have where chemicals are and what effect they are having on the charge, right, the electric side of our electrochemical gradient. And so we talked about diffusion and osmosis back when we were reviewing biology content. And so diffusion is basically describing that when we have a high concentration of molecules in one space, the kinetic energy of those molecules is going to cause them to basically push off of each other and try to spread out. And so in a scenario like this, where we're just considering the chemical portion of what we're drawing here, wherever there is a lot of some solutes, they are going to want to spread out to where there is more space. And so in the case of this cell that we've set up, we have a high concentration inside the cell and a low concentration outside. And so diffusion is going to, the chemical portion of this electrochemical gradient is going to drive these molecules to diffuse across the membrane to where there is more space until they reach equilibrium. So until there is like an even amount of them inside and outside. And then basically, once they have reached equilibrium, they will be moving inside at the same rate that they're moving outside, and there's no net movement. Right? They're just driven to move in the same direction. So that's just one half of the force that is pushing things around. The other thing to consider is the electrical part of this gradient, so the charge that these molecules are carrying. And so if these are sodium molecules, which are cations, right, they're carrying a charge of +1. Whenever they move into a space, they are bringing that charge with them. So if we draw back some of these sodiums that we had inside, Now we have charge to consider. And so inside of our cell, we have a higher charge, a more positive charge, and outside of our cell, we have a negative charge, since we have more sodium ions inside and less sodium ions outside. And so these two forces together can either both cause diffusion, like what we're seeing here, basically since we have a high concentration of these molecules inside, that's going to push them out. We also have an electrochemical gradient pushing them, or the electrical gradient pushing them because they want to go towards the opposite charge. And so when you combine those two ideas together, that is what is going to drive the movement of these molecules. And naturally, these molecules are constantly diffusing in and out of the cell, and some of these channels are always open. And so the net charge, based on where these different ions are, is the membrane potential. So when looking at things like our muscle contractions, which are stimulated by our neurons, or if we're talking about just action potentials in the neurons themselves, what happens is some signal opened another channel that was previously closed. Why can't I change my color? There we go. So say this channel was closed. Some signal caused it to open, which allowed for a bunch of molecules to move from one side to another in a way that they weren't before. And that changed the overall charge of this membrane. And so usually I'm kind of drawing the wrong direction for how this typically goes. But for an action potential, typically we're looking at sodium. And whenever a neurotransmitter or whatever other ligand that's going to cause this action potential, whenever that ligand bonds with a channel protein, that's going to cause it to open, and a bunch of that ion are going to flood into the cell. So in this case, we'll draw them going this way. They're all going to flood into the cell, which is going to change the electrical gradient. Now we're going to end up with a more negative charge inside the cell than we had before, and a positive charge outside. And once that charge reaches a certain potential, or sorry, I did that the opposite. Let me undo. Yeah. So since these sodium ions are carrying a positive charge, as they come into the cell, they're going to increase the charge inside the cell. And once that reaches a certain point, other membrane proteins that are nearby are going to continue to open because they are stimulated by that electrochemical gradient. And this is going to start a chain reaction where that continues to happen all the way down the plasma membrane. It's just going to keep going, keep going, keep going. And that is, in a nutshell, an action potential. The movement of these ions from the outside of the cell to the inside of the cell changed the electrical charge of the membrane, which is going to open up these other voltage-gated sodium channels. Which respond to that change in membrane potential. And so more sodium is going to continue to flood in over in these areas. And so that this change in membrane potential is going to continue like a domino effect all the way down the membrane. And then at the end, so like at the axon terminal of a neuron, where there may be some neurotransmitter being held in a vesicle waiting to be released. So at the end of that axon terminal, the change in membrane potential is going to cause those vesicles to release the neurotransmitter, which is going to cause another action potential on whatever cell it's responding to. So like in the case of muscles, it's going to cause an action potential down the sarcoplasm or the sarcolemma of the muscle cell, which is going to end up releasing calcium into the muscle cell. So the movement of ions carrying a charge changes the relative charge of the membrane, so making the internal component more positively charged and the external component more negatively charged, which opens up these voltage-gated ion channels, which causes a domino effect of more and more sodium rushing into the cell as that voltage reaches different portions of the membrane. And that will eventually lead to the release of some other molecule, like a neurotransmitter or calcium in a muscle cell. And there is a great video with like animations of this same process that I was drawing that I shared on Canvas. I guess before I go over there, is this part making sense? Did I do an okay job showing the step-by-step there to clarify your question?
Yes, thank you.
Yeah, cool. So I'm gonna switch us over to the Canvas site. Move this stuff out of the way. On Canvas in the module on muscle tissue. Here we go. So in module 8, in the useful videos for muscle, this video right here on electrochemical gradient does a great job describing that. Didn't mean to play an ad for you guys, but it's from Bozeman Science. So if you want to see that process in motion, I really recommend watching this video and trying out others. Like find creators that present this information in a way that makes the most sense to you. Like the Amoeba Sisters are typically more simplified looks at these processes, but they have really great animations. So I think it's the clearest example of these things. But some other creators like Bozeman Science is a very popular one. You see he has a lot of subscribers. He gets into the details of it, and that Crash Course Biology stuff gets into the details with great animations. It's kind of like a middle ground between those two. So check out the shared videos to see that stuff in motion, find creators that work best for you. Yeah, so that's how we stimulate the release of calcium all the way from a neuron to our muscle cells to allow our muscles to contract. Yeah, ExxonMobil, shout out the ads. Okay, what else? I really need you guys to point me in the direction that you want to go. I don't think it would be feasible for me to try to talk through all 4 sets of PowerPoint slides as review here. So need some sort of guidance to where you guys are struggling most.
Can we go over the structure of a hair follicle?
Sure. So the conceptual understanding here that I would be looking for is that the follicle itself is basically an extension of our epithelium, so the outermost layer of the skin. So it's extending or invaginating into the other layers of the dermis. Okay. And then basically all of the stem cell structures that we saw for epithelia are just contained into this roots here. Um, like I'm not going to get super specific with a question about a hair follicle. So I don't think it's all that relevant as far as applying it to something else. Right? But basically we have our stem cells that start maturing into thinner keratinocytes that are producing more keratin, and then those keratinocytes fully squish and die off the same way that we saw with our skin by the time they make it to the cortex, which makes up a majority of the mass or the area of the hair itself. They have their own papilla that contains the nerves and the arteries and capillaries that are going to service this each individual hair to keep these cells, these progenitor cells, these stem cells alive so they can continue making more keratinocytes that are going to extend the length of the hair. And then beyond that, just recognizing that hair follicles have a erector pili muscle, which is smooth muscle. And that's how our hairs stand up or change shape. And that helps with our ability to sense movement with the hair on our skin. And I guess I don't know if all, but definitely most hair follicles are going to have their own sebaceous gland that is depositing that gland secretions onto it. So sebaceous glands secrete oil, and then we have apocrine and eccrine glands, which secrete sweat. So again, most but not all hair are going to have an apicrine gland as well as a sebaceous gland that are secreting these two different types of secretions. And then hair that is in specific areas like the armpits and the anogenital region are where you find those apicrine glands that produce that really milky or protein milky sweat, which gets metabolized by the bacteria in those areas to produce body odor. So sebaceous glands are much more common. They secrete directly onto the skin with oil. Wait, sorry. I feel like I was saying conflicting info there. Didn't this say this was a sebaceous gland? Sorry, I'm going to come back to this in a second. Backing up, what I want you guys to recognize is there's an erector pili muscle. They often have glands that secrete directly onto the hair, and that the hair follicle itself is basically just an invaginated version of the epithelium. We have the same basic structures with very similar layers, keratinocytes maturing and squishing as they get more and more full of keratin. And then we have these two different gland types. I guess I was confusing myself with the directly onto skin versus not skin. So yeah, we do have sebaceous glands that secrete oil directly onto the hair, and then apicrine glands in specific areas like our armpits and anogenital region that only become active after puberty, which secrete a more milky, protein-rich secretion rather than that oily secretion, the sebum. Sorry that I kind of tripped myself up there. Was I able to save it? Did I clarify that? Or could I clarify something further?
It all makes sense to me.
Okay. What else?
One question I had was the steps in endochondral ossification. I feel like there was a question on the quiz about that, and it took me like 15 attempts to get.
Okay. And yeah, like just like with the last exam, I was saying the quiz questions are a lot more specific than typically what you're going to see on an exam question. Okay. So the general concept that I would be looking for your understanding on with endochondral ossification compared to intramembranous ossification is that endochondral ossification is basically creating bone from within. So there is a cartilage model, this hyaline cartilage model that creates the general shape of that long bone that it is going to create. We have these blood vessels that penetrate into the center of that hyaline cartilage model, and they will start to create bone. So replacing and breaking down the cartilage that is already making that model, replacing it with hydroxyapatite crystals and collagen, and then breaking down more bone so that eventually that bone is going to create and break down until it spreads out to the outer portions. And we're left with this big marrow cavity, this hole that is going to be filled with marrow later. Those blood vessels then penetrate into each of the epiphyses, where we will use the rest of this hyaline cartilage model to make more bone. And so at the end of this process, we're left with some cartilage at the in between the diaphysis and each of the epiphyses, and that is the epiphyseal growth plate that we see disappear over time as an individual matures. Right? With these images, right, they look like empty holes or broken, fractured bones if you look at the X-ray of an adolescent, and over time, that cartilage is being replaced with actual bone. So again, the concept here is that we're starting with a cartilage model. The blood vessels penetrate into the center and start creating the bone from within that model. And the diaphysis is created first, and then the epiphyses will be created after that, compared to, and this makes long bones, compared to intramembranous ossification, which forms flat bones, dermal bones. This type of bone growth is from the skin, basically, the epidermis, or from the dermis. So we have those stem cells that are going to eventually mature into osteocytes, and they are depositing hydroxyapatite crystals and collagen, creating their own matrix, surrounding themselves until eventually they're large enough that those matrices connect with each other and have now formed this one long composite bone. So again, endochondral ossification as a cartilage model, basically building the bone from within, and this makes long bones. So it starts by building out the diaphysis, and then we'll build the epiphyses. Intramembranous ossification comes from the dermis, or these bones are building from the dermis, from the deepest layer of the dermis, and that will eventually mature into a fully formed flat bone by simply creating matrix that extends out and eventually connects with each other. So we have bone connecting with bone, making one full individual bone.
Thanks, that helps.
All right, keep them coming. And Deacon, if you have more questions, please don't feel bad about continuing to ask them. I'd rather we be talking about stuff than sitting here silent.
What the Wednesday is our physiology test, right? Like lecture test?
Yes. Yeah, so
Okay.
next Wednesday is the lecture exam in lab.
Right.
And then the following Monday will be the lab exam, the muscle exam.
Okay. Then on that day, I'm just gonna prepare questions for your office hours on Wednesday just because I don't have many ready right now.
Okay, yeah, that's fine. Anyone else have any more questions? And even if you can't think of like a question, are there any topics or chapters that you find particularly difficult that I could talk about?
It's just a general question I have, like the exam. Is it gonna be similar to the first exam, or we expecting different ways?
Yeah, it should be very similar to the first lecture exam, very similar question types. And preparing for that exam should hopefully be the same, utilizing the learning objectives as a study guide and the suggested practice questions in that study guide should prepare you for any of the stuff that you would end up seeing on the lecture exam. So there will be a mix of some multiple choice, some matching fill in the blank type questions, some written response questions.
Okay, thank you.
Feel like I don't know what to say or do right now. You guys voted to have reviewed today. So I guess what can anyone share what they were hoping today would look like? Could I have set this up differently? Please feel free to jump in with questions or topics if you come up with them. The last three learning goals of chapter 10. Sorry, learning goals. So let me pull up So relate the types of muscle fibers to muscle performance, discuss muscle hypertrophy, atrophy, and aging. Okay, identify the structural and functional differences between skeletal and cardiac muscle cells. Identify the structural and functional differences between skeletal fibers, smooth muscle cells, and discuss the role. Cool. Okay. So for this one, for 10.8, we didn't directly address that or explicitly address, you meant the other sheet. Okay, let me pull up the other sheet. So just the general topics here, is this what you're referring to, Malika? Okay. So excitation, contraction, coupling, the contraction cycle and cross bridge formation, and steps involved in skeletal muscle contraction and relaxation. Okay. Yes. So I'm going to start with these two, because this one is just kind of an expansion on these ideas. So let's talk about the contraction cycle, which expands on the, what were we saying at the beginning of class? Action potentials. Okay. So the entire process that we learned about that leads up to the contraction of a muscle is going to start with the action potential from this motor neuron, then causing the release of a neurotransmitter. That neurotransmitter is going to cause an action potential on the sarcolemma of the muscle cell, which is eventually going to lead to the release of calcium, which is going to allow for the muscle to actually contract. And so I'll talk about all of those steps, but it is from the motor neuron all the way to the myosin head power stroke that is going to pull the thin filament towards the center, the M line. So sorry, trying to think of where to start. So we talked about action potentials, right? So the movement of ions into or out of the cell is going to change the overall charge on the membrane of that cell, which is going to open voltage-gated channels like this one. So we have the charge being measured of the internal environment of this cell by this little machine that they've attached to this membrane here, right? And our resting potential of most cells' membranes is -70 millivolts. And so whenever some signal is going to open another ion channel, that ion is going to flush into the cell, mainly sodium. So we can get specific and say this is going to open a sodium channel. That sodium is going to flush into the cell, which is going to increase the charge of the membrane all the way up to the positive end of the spectrum to +30 millivolts. And that is when the channel will close. And so as these sodium ions are rushing in, and that change in membrane potential is expanding along the membrane, more and more of these channels are going to continue to open. And so we kind of see that with this red line. So as sodium is flooding into the cell, the potential or the charge of the membrane is changing and is going to continue to change along the membrane. And so we see that red indicating the change in membrane potential continuing along the membrane until it's completely gone from this image. And that's basically going to continue all the way down, carrying that action potential down the T tubule, which is the extension of the sarcolemma down into the sarcoplasm, basically. So we have that action potential all the way along the membrane, down these T tubules until they reach the sarcoplasmic reticulum. The sarcoplasmic reticulum has its own voltage-gated ion channels like this that are going to open and allow calcium to flood out of the sarcoplasmic reticulum and into the cytoplasm of the cell, sarcoplasm. So that's what we see here. So backing up and talking about this again, we had an action potential that flooded down the membrane of this neuron, the axon, until it reached the synapse where the axon meets the cell that it is innervating. When it reached the synapse, it activated these vesicles or caused these vesicles to fuse with the membrane and release these acetylcholine neurotransmitters into this space. Okay, so the action potential flooded down, the vesicles start to fuse into exocytose, those acetylcholine molecules. Acetylcholine is going to bond with these ligand-gated sodium channels on the sarcolemma of the muscle cell. And that causes those channels to open, which is going to allow sodium to flood in, causing a new action potential on the muscle cell itself. That action potential continues all the way down the sarcolemma. Sorry, where's the image I had before? This one. All the way down the sarcolemma, down these T tubules until it reaches the terminal cisterns of the sarcoplasmic reticulum. And just like how the neuron released a whole bunch of acetylcholine when the action potential reached it, the sarcoplasmic reticulum is going to release a whole bunch of calcium when the action potential reaches it. And so that calcium floods into the muscle cell and is going to be able to interact with the troponin portion of the thin filaments. All right, so that is the activation portion. Okay. Action potentials cause the release of some ligand. That ligand was acetylcholine from the neuron. In this case, the ligand is calcium being released from the sarcoplasmic reticulum. And now this calcium is going to cause an interaction that allows our muscle to actually contract. So far, no contraction has happened. We're just talking about the signal that has been sent that is now going to allow for contraction to occur. The whole point of this was to get to the place where the sarcoplasmic reticulum released calcium into the sarcoplasm. Now that calcium is being released into the sarcoplasm, it can bond with troponin, one of our thin filament things here, thin filament proteins. So troponin is basically like a lock and key type protein that is holding these tropomyosin rope-like protein filaments onto the G-actin, which these myosin heads want to attach to. Sorry, I'm realizing I just used all of the vocabulary here in one sentence, and that's probably difficult to keep up with. So troponin is basically the gatekeeper, and it is preventing our active protein here, myosin, which is going to do the actual pulling. So troponin is like the security guard, the gatekeeper that is blocking myosin from being able to do what it wants, which is to attach to those ball-like globular actin molecules and then pull them. And so in order for this troponin to move the tropomyosin out of the way, it must bond with calcium. So we had that whole action potential signaling mechanism leading all the way to the release of calcium, sorry. We had that whole action potential, this excitation, contraction, coupling that all led up to calcium being released into the space within the cell, the sarcoplasm. And those calcium molecules are going to bond with troponin, which is going to move troponin out of the way and move tropomyosin with it. So now these ball-like G-actin molecules are available for myosin to attach to. So here's another look at that. So troponin right now is holding tropomyosin over these G-actin molecules, and myosin cannot attach. When calcium comes into the sarcoplasm, it is going to bond with troponin, and troponin is going to move and take the tropomyosin with it. So now all the active sites on this globular actin are available. And so myosin will happily bond with it, which is what it has wanted to do this whole time. So now that it's able to make this bond, it is going to release this ADP and free phosphate molecule that it's holding onto, which is going to cause the myosin to change shape, doing that power stroke. It's called a power stroke, causing the head to bend on the hinge, which pulls all of those thin filaments towards the center. So we basically have myosin heads facing different directions on each side of our myofibrils and the sarcomeres. Where's my good image of this? Here we go. Right. We have myosin heads facing in opposite directions that are all going to pull the thin filaments towards this M line, towards the center. Okay. So calcium allowed for troponin to move tropomyosin out of the way. Myosin is going to bond with the thin filament. And then once it releases ADP and phosphate, that is going to cause it to change shape, pulling those thin filaments towards the M line. That's what we see here. Right. So at rest, And then once calcium is present and that muscle, the myosin heads can do the power stroke, that is going to cause this contraction, the pulling of the thin bands towards the M line, towards the center. So we had that power stroke pulling that thin filament towards the center of the sarcomere. And then another ATP molecule is going to bond with myosin, which allows it to let go of that thin filament. And then hydrolyzing that ATP into ADP and P allows the head to snap back to where it started. And so then that process can repeat. If another signal comes in telling this process to continue, the calcium won't be reuptook, won't be taken back. The calcium will stay in the sarcoplasm, allowing myosin to continue to attach and pull, release, attach, pull, release until it runs out of energy. So there's all of that together. I think everything I said there addressed all three parts of this. Excitation, contraction, coupling, referring to the neuron and the muscle cell reacting to this excitation, to that action potential. The contraction cycle and cross bridge formation, which is that myosin head attaching to the thin filament and pulling it. And then steps involved in contraction and relaxation. So I guess another thing to talk about with relaxation is that the myosin head letting go of the thin filament does not mean that this muscle is automatically relaxed. Or I guess it is relaxed, but it has not gone back to its resting length. It takes some other force for those thin filaments to spread out again. And so for most of our muscles, that's just gravity, right? Like I'm contracting my bicep right now to flex my elbow joint towards me, and gravity will naturally pull my arm back down and allow that muscle to completely relax back to its original length. But that did require force. Without gravitational force present, my muscle would stay contracted unless I like pushed on it or something. So yes, this is one of the, I don't know how to describe that or what kind of adjective I was going to use there. But yeah, a question about this would be very similar to like the electron transport chain type question that you got with the unit 1 exam. So doing your best to describe the concept here with as many details as you can, knowing the step-by-step, or at least what's coming before the next thing, ultimately running from the start of this, which would be the action potential of the neuron, and ending with the myosin head pulling and then releasing the thin filament. And so again,
at what point does the calcium detach from the troponin?
at what point does it detach from the troponin? That's a good question. I'm trying to think of what would cause that. I don't know the direct answers to that question of how calcium lets go, or how troponin lets go of the calcium, rather. I imagine there must be another protein involved. I will have to look into the answers to that question. So I'm not just researching while you guys are sitting here waiting for me. But the answer is I don't know. I don't know why or when calcium is released from the troponin itself. I do know that the sarcoplasmic reticulum kind of reuptakes the calcium from the sarcoplasm when the action potential has left and the membrane has gone back to its resting potential. And so I guess maybe it's something to do with the electrochemical gradient itself, just naturally forcing the calcium back to the sarcoplasmic reticulum, but I don't know exactly. That's an interesting question. Yeah, it just says sarcoplasmic reticulum reabsorbs the calcium, but doesn't say how. Oh, there was a question last class that I looked into about the nutritional content of dark meat versus white meat, right? We looked at fast versus slow fibers. So slow fibers would look more red, they have more myoglobin to help resist fatigue and keep ATP production going normally. So they look more red to us, so that's our dark meats. And then fast muscle fibers look clearer or pinkish, and that's our white meat. The actual composition of each of these muscles is very barely different. There's more fat in, between these slow fibers, and thus there's a higher fat content. Barely, it's a very small difference, but there's a higher fat content in dark meat instead of white meat. And thus there is a higher calorie content in dark meat instead of light meat, because fat carries 9 kilocalories per gram, whereas proteins and carbohydrates carry 4. But other than that, there's no other nutritional impact of choosing dark meat versus white meat. Generally, the impact is the exact same, just a little bit more fat. Oh, and I was going to say, just like how I was suggesting with an acupotential, watching a video with animations is likely very helpful. Watching the step-by-step process of this excitation coupling reaction is likely going to help a lot with understanding this concept rather than trying to memorize steps or memorize facts. Getting to see it happen typically makes it make a lot more sense. So check out those videos. What's next, guys?
How much detail do we need to know about the sarcomeres?
You definitely don't need to know the names of all the lines and zones.
Okay.
So generally just that there are thin filaments and thick filaments, and that the thick filaments pull the thin filaments towards the center. It's basically all I care for you guys to know. So you don't need to know all these names, just that I'd rather you focus on the cross bridge formation and the actual method of contraction. So thick filaments pulling on thin filaments towards the center. And then for the structure of thin filaments, you don't necessarily care if you learn all of these different names, but you should recognize troponin, tropomyosin, and G-actin. Right? So the little globs, these balls are G-actin. That is what the myosin head is going to attach to. Troponin is the gatekeeper that is going to pull tropomyosin off of those active sites to allow for myosin to do that. Other than that, the rest of this is basically FYI for your info. I will not be assessing on those names. Okay. Generally just a difference in the structure of those joints. So they're both synovial joints, but the shoulder and hip joint are ball and socket joints, whereas the elbow is a hinge joint. Right? Elbow, knee, ankle, those are all hinge joints. So recognizing what kind of movement that allows for. And so our elbows, our knees, our ankles, they can really only flex and extend. There's not much side-to-side movement, very little side-to-side movement, if any. Whereas a ball and socket joint is triaxial and allows you to go in all different planes. So a lot more movement is available. And then a little bit about the structure that allows for that movement. Basically, the structure of the socket that allows for this head to sit in there and thus allows it to be flexible and rotate in all those different directions, compared to this knee joint, which has a lot more thick ligaments making up that structure, making it a lot more stable, but with less movement. So that general concept, a lot more movement, a lot more fluid, and less ligaments in a ball and socket joint. In a hinge joint, there is less movement because there's a lot more, or the ligaments involved are more numerous and thicker. So this is a more structurally sound joint, but it has less movement available.
I believe that the knee joint allows for a small amount of internal and external rotation too, right?
Yeah. Yeah. Yeah, none of these that they try, really none of these that I tried to define so specifically of only moving up and down, like that's not necessarily true, right? There's rotation a bit in most of these hinge joints. You can kind of rotate that bone within the joint to a certain extent. But in general, the noticeable movements do follow these axes. Right? And we even discussed, based on Constance's question, about what the other carpometacarpal joints look like, since this one is only talking about the saddle joint of the thumb, which looks very similar to a hinge joint. We have this circular rotating head sitting inside of a socket. We have this circular looking rotating head sitting inside of a socket, but because there's more space on the sides of that socket, it allows for a little bit of biaxial movement. So our carpometacarpal joints for our thumb doesn't just go down and up. I'm trying to do it without also bringing it in. It doesn't just go, there you go, flex and extend. It can also reach across for opposition. And even the carpometacarpal joints of our fourth and fifth metacarpal have a bit of that movement, but 2 and 3 only really flex and extend. They can't play opposition a whole lot. They have a little bit of it. What's next?
Sorry, is the allentoaxial joint the only pivot joint? We need to know.
You said the only pivot joint that we
Mhm.
need? Well, we have pivot joint as its own type. And then the ellipsoidal being separate from that. Maybe I misunderstood your question.
Oh, I was asking whether the allentoaxial joint is the only pivot joint in the body.
So sorry, again, I feel like I'm not maybe hearing you clearly. Right, the two examples of our pivot joint that we were given is the atlas axis, so between our C1 and C2, and then our proximal radial ulnar joint. So the joint where the radius and the ulna meet in the forearm, that is proximal, so closer to that elbow joint, which allows for us to rotate those two bones around each other.
What about the distal radial ulnar joint? Is that also a pivot
It's
joint?
staying static as I do this rotation.
Mhm.
Right, so the movement of the bone, the only like sliding or rotation movements, the actual joint is happening at the proximal point. But where these two bones meet at the distal point shouldn't actually be changing. they are staying relatively static to each other as they rotate. I believe, based on how this has been described and as I'm feeling it and doing it, that seems to make sense to me. I feel like your personal experience might show a difference with that if you know better than I do. Yeah, okay, maybe a better way to describe it or to make this make sense as I'm feeling it. Right, so when I rotate my forearm, and you guys could try feeling this at home too, like you hold on, if you try to feel and hold on to the ulna of your forearm while you rotate it medially, you can feel that the bone, like the olecranon, this entire ridge of the bone here, none of that is changing position as you rotate. It's staying in the exact same place, but the radius is moving. It is changing position. And now if we do the same thing at the distal part, if you try to hold your ulna at your wrist and do that same rotation, you can feel the bony markings of the bone moving. So they are moving together statically at the distal part, but at the proximal part, the ulna is staying still while the radius rotates around it, pivots around it. Does that make sense? Okay, gonna send out this quick poll to see if it's worth sitting around and waiting for more questions, or if you guys feel like you're all out of questions, then we should just end class. Okay. Still waiting on a couple students to respond, but as that's going, I think if we get the opportunity to do a unit 3 review, I am going to structure that differently to try to avoid this, how awkward this class meeting has felt, at least for me. This is probably one of the most awkward Zoom calls I've been on. So in the future, I will set up a survey with like numbers that correlate with the learning objectives, or numbers that correlate with the topics mentioned in the Unit 3 exam study guide. And I'll ask you guys to respond to that survey before our class meets for that review day. And then I'll just use those responses to guide this. So that way everybody is giving some input into which topics have felt the most difficult. And that will give me some idea of generally how a class time is going to be structured before we come to the Zoom meeting, rather than solely relying on people to volunteer with questions and topics. So hopefully that works out better. And I'm sorry that this was a bit slow and maybe a bit disjointed, but I hope you guys got the help and the answers that that you needed leading up to Wednesday. I will have office hours next week. Of course, I always I respond to emails and messages is what I'm trying to say. So send me your questions if you come up with them. But other than that, I hope you guys have a great weekend, and I will see you next Tuesday. There is no school on Monday, so no lab on Monday. I will see you next Tuesday to start Unit 3.